Today is the 10th November 2020, and it's my pleasure to have Brian Byrd from the USA, University of Michigan Medical School, to join us. So Brian has investigated hypertension, a leading risk factor for death and disability, via novel phenotyping methods. His laboratory is conducting clinical studies of novel ex vivo assays of mineralocorticoid receptor activation using urinary extracellular vesicles and their mRNA cargo as a window of these receptors' ligand-activated transcription factor activity. So today, his talk is titled Extracellular Vesicles as Novel Biomarkers in Hypertension. So please welcome Brian.
Thank you. I'd like to thank Carolina for that introduction and the kind invitation to present our work as part of this very creative project, WebEV. I'm delighted to be presenting from Ann Arbor, Michigan, which is the home of the University of Michigan, where I'm part of the faculty. And I'm showing some photos that I've taken on campus to show you a little bit about what the university looks like. What I love about this particular invitation to speak is that the talk is free for anybody to hear. Anybody can be in the room virtually speaking, irrespective of boundaries or borders or institutions. And it's just about who wants to hear a certain set of ideas. I find that very powerful. And it means that the audience can include anybody anywhere. But if you're watching, I think you probably have some interest in extracellular vesicles, which I'll also call EVs. And for that reason, I know we have something in common. These are my disclosures, which include some funding sources for different projects, as well as a provisional patent related to some of the work that I'll talk about during this talk today. Because the audience likely has very eclectic interests beyond our shared interest in EVs, I want to start by orienting you to the areas of knowledge that I draw on in my work. I'm a cardiologist, and I'm trained in clinical research with a focus on biomarkers. And in view of that background, I'm interested in 3 topics, each of which is relevant to this talk. The first thing I tend to think about a lot is high blood pressure, which is the most important health problem in the world by at least 2 or 3 metrics, and I'll share more on that in a moment. The second thing I think about is clinical pharmacology, which is the study of the effects of medications in humans. And I did a fellowship in clinical pharmacology. And within the very expansive topic of high blood pressure, what interests me the most is the effects of medications used to treat high blood pressure. So that's what links these interests. Many of the medications that we use to treat high blood pressure act on the kidney directly or indirectly. And as a rule, we don't really customize the treatment of high blood pressure to the patient. Instead, we start with one of 4 drug classes as the initial treatment of high blood pressure. And those could be picked more or less at random. There isn't really, the guidelines don't prioritize one over the other. And this approach is recommended by the guidelines despite some known differences in how people respond to them that involve some challenging tests to figure out who would respond how. And it's not very pragmatic to do that for the very large number of people who have high blood pressure. So it's pragmatic to treat everybody essentially the same initially with one of 4 blood pressure medicines, each of which is considered to be reasonable. And there are times when that approach doesn't work well. And that's when we need some customization or personalization of treatment to the patient. And that brings me to the third thing I think about, which is urinary extracellular vesicles, which have potential to help customize the treatment of high blood pressure in several ways that I will go on to discuss. So the direction of this talk will be from very big to very small. We'll start with the biggest health problem, high blood pressure. I'll tell you enough about it so you can understand why I'm interested in urinary EVs and what problems EVs might solve in the world of high blood pressure. And as we move to the very small scale, the nano scale, we'll talk about some studies of EVs in high blood pressure. So let's talk about how things look for the problem of high blood pressure today. High blood pressure, which is also called hypertension, is a growing problem around the world, mostly due to aging. This figure shows the growth in the number of adults with raised blood pressure as defined as a systolic blood pressure, or top number, if you will, greater than or equal to 140 millimeters of mercury, or a diastolic blood pressure greater than or equal to 90 millimeters of mercury, the diastolic being the bottom number. And as you can see, it has been about 14 years since we passed the 1 billion mark in terms of people with high blood pressure above that 140/90 threshold. And the threshold for the diagnosis of hypertension was lowered in 2017. So the number of people who meet the definition for hypertension now is much larger still by a contemporary definition. Nearly 10 million deaths a year can be linked to hypertension. And hypertension causes not only deaths, but a great deal of disability in the form of strokes. In fact, hypertension is the leading risk factor for reducing disability-free longevity. And high blood pressure is projected to still be the leading cause of premature death in 2040, unless we bend the curve in some dramatic way. So I don't think it's an exaggeration to say that high blood pressure is the most significant health problem faced by people and by health systems. A lot of people who have high blood pressure do not get their blood pressure under control. There are about 108 million US adults with high blood pressure, and 87 million of them meet criteria to be treated with medications as opposed to just lifestyle improvements. 61 million patients have uncontrolled high blood pressure, and for complex reasons, about half of those people are not treated at all. The other half are treated with medications, and that's 31 million, and still don't have good blood pressure control at the threshold of 130/80. And a fair fraction of them don't have blood pressure control even at the higher level of 140/90. So 17 million people have frankly uncontrolled hypertension, and more, 31 million, have treated but at least modestly uncontrolled hypertension. So the situation regarding blood pressure control needs improvement, and biomarkers may have a role to play in that. Okay. In hypertension, as I mentioned, we've taken a pretty pragmatic approach to treatment. And we've taken a pretty pragmatic approach to biomarkers, which, as you know, are a defined characteristic of a normal biological process or pathogenic process or a response to an exposure or intervention. And we can use a framework provided by the FDA NIH Biomarker Working Group to think about how biomarkers could be used in the realm of hypertension. You could develop a biomarker to try to estimate the risk of developing hypertension, or maybe more rationally, the risk that you would develop a certain type of hypertension, like the type that is very damaging or causes stroke. You could try to confirm the presence of a particular subtype of hypertension, and I'll bring out a certain subtype that is of great interest called primary aldosteronism in a moment. You could monitor the status of organ injury in hypertension. You could try to prognosticate who might go on to have a stroke or other bad event. You could look at predicting the response to special medications like the ones I'm going to talk about today. You can try to measure the pharmacodynamic response to the medication that has occurred. I'm going to talk to you about a receptor antagonist today, and you might be able to try to infer to what extent that receptor is in fact antagonized in the person. And then you could also try to develop biomarkers related to safety and toxicity. So over time, we've introduced some molecular biomarkers into our guideline-based care of hypertension. Molecular being used in a fairly broad sense here. I'm not referring just to nucleic acids. In fact, that is not in play in terms of guideline-based care of hypertension right now. I highlighted the current generation of guideline-based biomarkers recommended in the 2017 American Heart Association and American College of Cardiology hypertension guidelines. And the biomarkers that have, in a sense, a molecular aspect are shown in bold. And a characteristic of the current generation of clinical biomarkers in hypertension is that they were developed a long time ago, and they're really quite primitive by the standards of modern science. It's important to generate new leads on novel biomarkers that might allow us to characterize high blood pressure and its effects on the body and its causes in a more refined way than we can do with these old markers. So as this group knows, by the 1970s, several groups studying the ultrastructure of the cell noticed in their electron micrographs vesicles near the cell surface or budding from the cell surface. And this is a 1971 electron micrograph of golden alga, and you can see vesicles budding from the cell surface at the spots that I've highlighted with blue arrowheads. And of course, since that time, we've come to define these extracellular vesicles as nano-sized particles with a lipid bilayer, and the secretion of these vesicles is highly conserved. In fact, I've not been able to find an example of a cell in nature that doesn't produce extracellular vesicles. If you know of one, tell me on Twitter or email me or tell me at the end of the talk. I'd be interested to know about that. But it's at least a highly conserved process. And there are at least two pathways of release. So it's not a simple biogenesis concept that can be conceptualized as one pathway. This is a figure showing some of the different kinds of extracellular vesicles, a figure from a paper that I published recently with Ute Erdbrugger and colleagues. And our ability to see and characterize extracellular vesicles has progressed, obviously, since 1971, and now they can be imaged with extraordinary detail and resolution. And on the left is a modern cryo-EM image of vesicles, and on the right is a leukemic monocyte with budding vesicles visible in the cell surface and near the cell surface. And with these technological advances in our ability to study vesicles, there's been a corresponding increase in interest in how these extracellular vesicles might be used to help patients. And I was invited in 2018 to recapitulate the more clinical work presented at the International Society for Extracellular Vesicles meeting in a talk at the end of the meeting. And then I was asked to publish the analysis as a snapshot of the field. And so we classified abstracts using inductive logic, basically finding that the most common themes for studies that had a clinical or clinical sample aspect were oncology or the use of clinical samples to develop new methods. And at the same time, we also saw that there were cardiovascular medicine applications people were interested in rheumatology and endocrinology and infectious diseases, lots of different things. And we found that people from around the world were working on this. I gather there are more people working on it now than in 2018. And the other thing I would notice is that from the abstracts that disclosed their N, we analyzed the average N, if you will, and it was very small, consistent with a field that's just getting started. Sensibly enough, people will generally not give large amounts of money to do a big cohort study if a field is fairly nascent. And I think that's the situation that we found ourselves in 2018. And so like others, we've been thinking about how a new generation of biomarkers might change the care of patients, and in particular, in my case, the care of patients with hypertension. Generally speaking, these are the roles that I just went over that for potential biomarkers in hypertension. But let's talk about a specific concept of a biomarker. In order to understand how a specific biomarker might play a role in the care of patients with hypertension, I need to introduce a really fascinating hormonal cascade that was absolutely essential to the survival of humanity before the mining of salt, sodium, started thousands of years ago. And now this hormonal cascade may be best thought of as vestigial in modern life when we eat a lot of salt, and it may in fact be harming us. This cascade is called the renin-aldosterone cascade. Recall that minerals such as sodium are minuscule, and they pass freely from the blood into the urine. And they would very rapidly leave the body entirely if there were not a system that reabsorbs sodium back into the urine, back from the urine rather, into the blood. And of course, the renal tubule is the anatomic location of that reabsorption, but let's think about what regulates that reabsorption. Obviously, we need the right amount of sodium in the body for proper cellular physiology. And the proper amount of sodium to reabsorb is a question of homeostasis. In other words, it's determined by how much sodium one is eating. What goes out should match what goes in, more or less. And a proper set point is important so that you have the right amount of sodium and water that comes with it during reabsorption to maintain proper fluid volume in the circulation and proper pressure, blood pressure in the circulation. And that's where this hormonal cascade called the renin-angiotensin-aldosterone system comes into play. During the prehistoric struggle to find resources, the amount of sodium that you might take in a meal or in a day even might be just what was in the ashes of wood that you use to cook a sweet potato. Or maybe you get lucky and you catch a bird and there's some sodium and some water in that bird. In that setting of truly minimal sodium intake, the perfusion pressure sensed by the portion of the kidney called the juxtaglomerular apparatus will be low because the volume in the circulatory system will be low and the pressure will be low. That low perfusion pressure stimulates the secretion of the first hormone in the renin-angiotensin-aldosterone system, that's renin. That leads downstream to the secretion of aldosterone from the adrenal gland that sits atop the kidney. Aldosterone stimulates a cytosolic nuclear hormone receptor called the mineralocorticoid receptor, regulating the presence of a sodium channel on the apical or urine-facing surface of epithelial cells in the distal tubule of the kidney. And that sodium channel is called ENaC. We'll talk more about that. So ENaC is this regulated channel that reabsorbs sodium in proportion to the amount of sodium you need to reabsorb based on how much you're eating. Eat more sodium, the renin-angiotensin-aldosterone cascade gets shut down, and the kidney permits a correspondingly greater amount of sodium to leave the body in the urine. That's the expected state in modern humans. People eat a lot of sodium, suppressing the renin-angiotensin-aldosterone system. So as I said, in a sense, the biological cascade of hormones called the renin-angiotensin-aldosterone system has been rendered somewhat vestigial by the huge amount of sodium that people eat. It's just shut down in many people. We've known since the 1950s that a disease called primary aldosteronism causes inappropriate secretion of aldosterone in people who are eating a high salt diet and in whom the earlier portions of the hormonal cascade are suppressed. This is due to abnormalities in the adrenal gland itself, and it is not a well-tolerated situation physiologically. In animal models of the consumption of a high salt diet and activation of this mineralocorticoid receptor with compounds like aldosterone, this hormone, there is fibrosis of the kidney, fibrosis of the heart. It's very damaging. And inappropriate aldosterone secretion in the setting of low activity of the earlier portions of the renin-angiotensin-aldosterone system means that the blood pressure medications that interrupt the earlier parts of that cascade, which are shown in this figure from the New England Journal, those blood pressure medications will be ineffective. The only medications that will block the injury, the tissue injury that comes from high salt diet plus aldosterone secretion would be drugs like mineralocorticoid receptor antagonists, which block the effects of aldosterone on the mineralocorticoid receptor. Those include spironolactone and eplerenone. So it turns out in some people who are secreting an inappropriate amount of aldosterone, it matters which drug you use. The other drugs will not be as effective. And even if you can control the blood pressure, people will still be at risk for the tissue injury that comes from inappropriate aldosterone effects on the body, which include increased risk of stroke, increased risk of atrial fibrillation and chronic kidney disease compared to people who have high blood pressure without inappropriate aldosterone secretion. Primary aldosteronism, when it was first discovered in the 1950s at University of Michigan, was thought to be a rare problem. However, this is a paper from July of 2020. It turns out that if you try to suppress the renin-angiotensin-aldosterone system by giving oral sodium to people with normal blood pressure or untreated mild high blood pressure, you'll find some of the people have unsuppressible aldosterone production as represented by the amount of urine excreted in 24 hours in the urine. I'm sorry, the amount of urinary aldosterone is secreted in 24 hours. And what you can see is in this top graph, the number of people who have above a threshold amount of aldosterone production despite an attempt to suppress aldosterone is, that's an arbitrary threshold, but you have some people with untreated normal blood pressures who exceed this threshold. And you have more people who have stage 2 hypertension, which is quite high blood pressure. And then you have even more people in whom the treatment of hypertension is stymied by resistance to the medications. Those patients very often have a lot of aldosterone production. And as you can see in the lower left-hand corner, there's just a general shift in the amount of aldosterone being produced in people as their blood pressure is higher. And a similar finding is recapitulated in the lower right-hand corner. So as I mentioned, aldosterone is responsible for activating the mineralocorticoid receptor. Aldosterone is a steroid hormone, and it diffuses freely through the cell membrane where it binds mineralocorticoid receptors in the cytosol. Those receptors then dimerize and translocate to the nucleus where they bind to hormone response elements in the DNA, regulating the transcription of certain genes, including one called SGK1, one called GIL-Z, which has a transcript TSC22D3, and then ENaC, which is that sodium channel I mentioned. Then the net effect of this changed program of gene expression is increased expression of the epithelial sodium channel on the apical or urine-facing surface of these epithelial cells in the kidney, allowing sodium to be reabsorbed through the cell and back into the blood. That's fantastic if you're in a salt-deprived environment. But again, animal models and studies of inappropriate aldosterone secretion in humans suggest that mineralocorticoid receptor activation is a contributor to high blood pressure, including difficult to treat high blood pressure, organ fibrosis, problems like atrial fibrillation, and stroke if this biology of mineralocorticoid receptor activation is prominent while you're eating a lot of sodium. It's frankly inappropriate, but it does happen. Further evidence that mineralocorticoid receptor activation leads to treatment-resistant high blood pressure comes from a study called the Pathway 2 trial done by Brian Williams and colleagues in the UK. They studied patients whose blood pressure remained too high despite treatment with a regimen that included 3 different classes of blood pressure medicine. So this is really tough to treat high blood pressure. And in a randomized crossover design, comparing the effect of adding different fourth medications, a drug called spironolactone, which is a mineralocorticoid receptor antagonist, was the drug that best reduced blood pressure, which was measured at home in this particular study. We have additional direct evidence that pathological activation of the mineralocorticoid receptor causes health problems, not just resistant hypertension. There are data, these are data from the Rawls clinical trial, which compared the use of a mineralocorticoid receptor antagonist or placebo in patients with severe heart failure. And as you can see, over the course of 3 years, the probability of survival, illustrated on the y-axis, was higher in patients who were treated with mineralocorticoid receptor antagonists. So you can see that activation of the mineralocorticoid receptor, when inappropriate, can cause health problems. And antagonizing the receptor with FDA-approved drugs can be the key to improving difficult to control high blood pressure and preventing deaths in heart failure. So this is a really important mechanism, a really important receptor in human biology, even if in normal daily life, the renin-angiotensin-aldosterone system is shut down by all that sodium that we eat. It's usually quite quiescent. There are people in whom it is an inappropriately active system to the detriment of health, particularly the aldosterone component of the system. So it's obvious then that people should be prescribing one of the FDA-approved drugs that blocks the mineralocorticoid receptor in order to treat problems like treatment-resistant hypertension. And this graph shows that about 10% of patients with resistant hypertension actually get these drugs. The line that is pink and says aldosterone antagonists is the line representing mineralocorticoid receptor antagonists. It may be difficult to see, but it's essentially below the 10% line. So of people who appear to be reasonable candidates for these medications, really fewer than 1 in 10 are treated with them. What about in heart failure? I just showed you that these drugs save lives in the setting of heart failure. And again, clinicians are not using these medications. About a third of the time, people get these medications when it appears to be appropriate. And that is actually getting worse over time. What's going on? So in a survey of clinicians asking why these inexpensive, quite old generic medications that are very useful are underused, the most common reason given was fear of side effects. And that concern cannot be dismissed because the mineralocorticoid receptor not only regulates the excretion of sodium from the body, but also the excretion of potassium. And fatal high serum potassium is a feared complication of these medications, the mineralocorticoid receptor antagonists. So there's an unmet need for biomarkers of mineralocorticoid receptor activation. It would be helpful if clinicians could better assess the risk-benefit ratio of using one of these medications in the patient that is in front of them, so they can be reassured with respect to whether or not they're causing more harm than good using these. Currently, we have no biomarkers that are a direct readout of the mineralocorticoid receptor, and I've listed some possible surrogates people have explored, but nothing has emerged as useful or usable in the clinic. In the lab, we knew that Mark Knepper and his colleagues had discovered in 2004 that extracellular vesicles present in the urine shuttle a variety of proteins relevant to the regulation of blood pressure. And that includes things like the angiotensin converting enzyme, the alpha, beta, and gamma subunits of ENaC, the epithelial sodium channel that I mentioned earlier, and other molecules relevant to sodium reabsorption. And Mark Knepper made a very elegant illustration showing the biogenesis and secretion of one type of extracellular vesicle exosomes from tubular epithelial cells into the urinary space, which I'll show here. And my good friend Matt Luther's group at Vanderbilt University took that discovery and moved it in the direction of translation toward a biomarker of ENaC activation. And what he found was that in 14 mildly hypertensive patients placed on a low sodium diet to stimulate aldosterone production by stimulating the renin-angiotensin-aldosterone system, and then in whom the system was later suppressed by a high salt diet and a crossover design, the ENaC gamma subunit, so the gamma subunit of the ENaC protein in urinary extracellular vesicles, was upregulated by the high salt diet. In addition, when he infused aldosterone versus vehicle into volunteers, he found that the aldosterone level correlated with the gamma ENaC peptide in the urinary EVs. And this was done using a technique called multidimensional protein identification, or MUDPIT. And there were some other markers that he looked at that were not different, but some evidence that the urinary exosome shuttled or EV-shuttled ENaC protein is being regulated in a way that's correlating with biology of interest to people like me who are interested in high blood pressure. So we became interested in a more direct readout of mineralocorticoid receptor activation, not the proteins that are being regulated downstream, but the ligand-activated transcription factor activity of this nuclear hormone receptor itself, the mineralocorticoid receptor. So we became interested in whether or not urinary EV-shuttled mRNA could be a more direct readout. We knew that in animal models, if you wanted to detect the activation of the mineralocorticoid receptor, the answer was to remove the kidney and to assay for certain genes that were known to be regulated by activation of the receptor. And we were wondering whether we could find those genes in urinary extracellular vesicles. Promising from that perspective was that approximately 13,500 protein coding genes had been discovered by Miranda et al. In their assessment of total RNA sequencing of urinary extracellular vesicles from a healthy volunteer. These were isolated by ultracentrifugation of a large volume of urine. So the approach that we began to develop, the strategy we began to develop for a biomarker of mineralocorticoid receptor activation per se, not the downstream protein regulation, but the activation of the receptor, was based upon this insight that the RNA might be present in the urinary EVs, the RNA that would reveal the ligand-activated transcription factor activity of the receptor. So the proximate event indicating MR activation is changes in gene expression, as I mentioned, SGK1, ENaC genes, TSC22D3. And we knew that urinary extracellular vesicles contain molecules from renal tubular epithelial cells due to work by Mark Knepper and others. And we knew that EV-enclosed RNA in human urine could be assayed due to work by Miranda and colleagues and others. So our hypothesis was that it was possible to detect mineralocorticoid receptor activation using mRNA shuttled in urinary extracellular vesicles that would be in contact with these tubular epithelial cells where this biology is of interest is happening. But a preliminary question was, can urinary EV-shuttled mRNA be used as a biomarker of any process that occurs in the kidney at all? Is there any relationship between the mRNA content of urinary EVs and the cells in the kidney? And really, we hadn't seen any analysis that would look at that in any comprehensive way. So we undertook to make a comparison. We compared the mRNA-seq transcriptome of urinary EVs from the healthy male volunteer that I mentioned a moment ago, sequenced by Laliata Russo's group, Miranda and colleagues at Harvard. And we compared the transcriptome to mRNA-seq transcriptomes of a series of human kidney cortex samples that had been sequenced post-mortem for the NIH-funded GTEx consortium. And along the x-axis is the series of male kidney cortex samples. We knew that based on the publication from the Miranda group, we knew that the volunteer who provided that urine was male. So to simplify the matter, we selected male GTEx consortium samples to compare with. On the y-axis is the correlation coefficient for normalized abundance of gene transcripts. The pink bars represent genes previously identified in the GTEx consortium by another group as kidney-enriched genes, and the blue bars represent genes previously identified as ubiquitously expressed genes. And we made that distinction because the bioinformatics expert who joined us in this analysis pointed out that if we did not separate ubiquitously expressed genes, we could end up with the wrong impression driven by ubiquitously expressed housekeeping genes. And so I think that was very smart. So as you can see, despite the fact that the urine came from a different donor compared to these kidney cortex samples, and there's a living person who's healthy versus post-mortem samples, the correlation between the kidney cortex transcriptome and the urinary extracellular vesicle transcriptome is still quite strong, especially for kidney-enriched genes. And we found that reassuring with respect to the potential for the mRNA in these urinary EVs to be a biomarker, for example, of MR activation. We collaborated with a colleague at University of Michigan, Scott Hummel. He had asked patients with pre-hypertension, so hypertension not mild enough to really meet the definition of hypertension, to consume a low salt diet for 4 days. And that would activate the renin-angiotensin-aldosterone system and activate mineralocorticoid receptors. And then he infused saline, so salt water, after 4 days to suppress the renin-angiotensin-aldosterone system. And he had collected blood and urine samples before he did the saline infusion and after he did the saline infusion. And as expected, during low sodium diet, the amount of sodium being excreted relative to the amount of creatinine in the urine was low. And then after sodium loading, it went up a lot. That's what you'd expect. That's just a little bit of reality testing to make sure that worked and that people actually consumed the diets. Then in my laboratory, we developed some novel ex vivo assays that were focused on detecting mineralocorticoid receptor regulated genes, as well as control genes that we expected not to be regulated by the mineralocorticoid receptor. So our primer design relied on exon spanning primers. We also used locked nucleic acid fluorescent probes to increase the sensitivity and specificity of the assay. And because we expected small amounts of RNA, we used pre-amplified qPCR protocols. And we used a small aliquot of urine that had been previously stored at -80 degrees. Because Dr. Hummel had done this study using standard methods, we had also things like the serum aldosterone concentration and how much renin was in the body, how much sodium was being excreted in the urine. I showed you that a moment ago, how much creatinine was being excreted in the urine, et cetera. And the genes that we decided to assay for that we thought of as mineralocorticoid receptor regulated genes included aquaporin 2, the 3 subunits of the epithelial sodium channel that are expressed canonically in humans, SGK1, and TSC22D3, which I mentioned before. In addition, we looked at some control genes we expected not to change with modulation of the renin-angiotensin-aldosterone system. One of those was NR3C2, which is the mineralocorticoid receptor itself, and then other things like uromodulin, which is the most abundant protein in normal human urine. So we designed and evaluated 25 probe-based qPCR assays for targets in 11 genes that are either MR regulated or control genes not thought to be regulated by MR. The reason we had more probe-based qPCR assays compared to genes was because we expected fragmentation of the mRNA, and we wanted to design for the, when possible, for the possibility that the fragmentation might affect our results. So we would assay in some instances at different parts of the gene. We applied the assays to RNA extracted from cell-depleted urine samples after enriching for urinary extracellular vesicles. We used the Norgen kit to use charge affinity to attract vesicles to a silicon carbide solid phase. We lysed those vesicles, and then charge attraction allowed the RNA to bind to the silicon carbide solid phase, and then we eluted the RNA. In the end, we had 858 sets of technical triplicates, which is 2,574 qPCR results, of which we deemed 658 sets to pass quality control filters at the triplicate level. When we looked at this initial question of how do these assays perform, I mean, we were trying to do something quite novel, and we wanted to make sure that the general performance characteristics of the assays made sense. So we looked at the technical replicates, technical replicate 1 versus 2, 1 versus 3, 2 versus 3. And what we found was very high correlation between the technical replicates, which we found reassuring. Then we looked at the question of how highly correlated are two different primer probe assays within the same target gene. And we found that the answer in terms of CT value is not as highly correlated as technical replicates, of course, but still generally highly correlated with some variation for some of the genes. And I think we have some evidence since then of the bias in the representation of mRNA that ends up in urinary extracellular vesicles, which may explain some of this. There could be some splice variants that explain some of this, or imperfect assays. There's different possible explanations, I think, that still need to be looked at. The next thing we looked at is whether or not sodium loading did indeed suppress the renin-angiotensin-aldosterone system as expected. And we did find that the first component of the renin-angiotensin-aldosterone system was suppressed, as well as the terminal component, the aldosterone. So that was exactly what you would expect with sodium loading. And of course, as I mentioned, the urinary sodium excretion increased after sodium loading, which is perhaps obvious that that would happen to maintain homeostasis. And creatinine was not different. So in terms of our novel assays, 5 of the primer probe pairs for genes that included the alpha subunit of ENaC, 2 assays for the gamma subunit of ENaC, an assay for SGK1 and an assay for TSC22D3, changed with sodium loading in the direction you would expect. In other words, toward higher CT value, if this is the direction you would expect if this were a set of biomarkers of mineralocorticoid receptor activation. Although these are acting in concert to regulate the epithelial sodium channel's presence on epithelial cells of the kidney, the reviewer of our manuscript asked that we use Bonferroni correction, which is really appropriate when these things are independent rather than networked. But we did do that, and I can say that one of the assays still withstood Bonferroni correction in terms of the significant change. And that was one of the SCNN1G assays. This work is published. I didn't put the citation here, but I think on some other slides it may be there. This is published in Circulation, Precision and Genomic Medicine. So The other thing that we found is that one of the assays for mineralocorticoid receptor itself also changed in the direction of a higher CT value. The next thing I would point out is the very same 5 assays that changed with sodium loading also correlated in the direction you would expect of a biomarker of mineralocorticoid receptor with aldosterone concentration in the serum. We also looked at whether this network of genes that's regulating sodium reabsorption correlated in the expected direction with urinary sodium excretion in this study, and the answer was yes, it did. As I mentioned, there was no difference in urinary creatinine at baseline versus after sodium loading. What we did in order to normalize these assays was we looked at samples of urine that had been, aliquots of samples of urine that had been collected over a period of hours in order to mitigate against some of the differences that occur in urine. I'll talk more about that in a second. In conclusion, the transcriptome of urinary extracellular vesicles in humans shows similarity with the transcriptome of human renal cortex. Transcripts of MR target genes were detected in cell-depleted urine after enrichment for extracellular vesicles. And a low sodium diet was accompanied by increased abundance of mineralocorticoid receptor regulated genes. And the abundance of mineralocorticoid receptor regulated gene transcripts correlated directly with serum aldosterone concentration and inversely with urinary sodium excretion, as would be expected of a biomarker of mineralocorticoid receptor activation. We're not the only people who are interested in this topic. Silvia Monteconi in Italy has studied patients with primary aldosteronism looking at plasma EV-RNA, and she compared 12 people with primary aldosteronism or inappropriate secretion of aldosterone with 12 people with essential hypertension. She found that in the plasma, there were 2.2-fold higher EV concentrations in patients with primary aldosteronism compared with patients with essential hypertension. And by microarray, she found 15 mRNA targets that were upregulated and 4 targets that were downregulated in primary aldosteronism. So this may be some early signal that there can be some value of plasma EV-shuttled RNA in primary aldosteronism. In addition, a closely related concept has been explored by a group at the University of Virginia. Gildia and colleagues looked at people who had previously been phenotyped for a phenotype called salt sensitivity. And that's basically a big increase in blood pressure when people eat sodium. That's not present in everybody, but some people have it. And in order to discover that a person has that problem, you have to give them the sodium, which is inconvenient as a test. They were interested to know whether microRNAs in urinary EVs might be revelatory with respect to whether people have this phenotype. And they did find 194 microRNAs that were above background in the urinary EV of these participants, of which 45 were significantly different by salt sensitivity phenotype, suggesting that maybe there's potential to develop a predictive biomarker of salt sensitivity. So to go back to this framework for a second, we think these biomarkers that we and others are developing have potential to impact the diagnosis of subtypes of hypertension, in particular primary aldosteronism, to monitor how organs are being injured by inappropriate aldosterone secretion, to determine patients' prognosis potentially, and to predict the response to mineralocorticoid receptor antagonists to help clinicians judge whether they can safely use those drugs or whether at least the safety is balanced, safety considerations are balanced by a good reason to expect a big benefit. And then also we think that it will be useful to measure mineralocorticoid receptor activity as a readout for how effectively one has suppressed the mineralocorticoid receptor activity in people. These are new concepts. We're excited to continue evaluating what's possible. Of course, there are challenges. A problem that arises when working with urinary EV biomarkers is how to optimally normalize the signal. And I would refer you to a recent paper by Dr. Peter Ewan, in which he found in rats that TSG101 is an excellent surrogate for small EV secretion, and its excretion mirrors the diurnal variation in EVs. The paper has a lot of important insights, and I would encourage you to take a look at that if you find this topic interesting. We've also written a paper about urinary EV biomarker normalization with a focus on RNA shuttled in EVs. If you'd like to read some of the special considerations that apply, have a look at this paper. I'll mention a couple of them. There are at least two potential sources of confounding when looking at urinary biomarkers that are EVs shuttling RNA. One is general changes in the composition of urine. Urine is a changing fluid biomatrix, or biofluid matrix anyway. The changes that occur when people drink water or sweat are going to have broad effects on various analytes concentration. So normalizing for that in some way is rational. The other thing is there may be disease states that increase the number of vesicles that are secreted or the amount of RNA that is shuttled in the vesicles in a unit period of time. That's another thing that may be important to normalize for, depending upon exactly how you are what you're measuring and how you're measuring it. So we concluded that pending new investigations, probably the most reasonable molecule, if you want to use a molecule instead of integrating over time with 24-hour urine collection or timed urine collection, would be urinary creatinine. And we said that despite our intuition that it might not be the optimal choice for any particular application, but it's probably the best first approximation, broad choice that would apply to many things. At the end of the day, we think a bespoke selection of an mRNA molecule expressed by the cells that have the physiology or pathophysiology you're interested in, and in cells and molecules that are not dysregulated by the pathology you're interested in, we think that probably has theoretical advantages over creatinine. One is that if there is a global export increase in terms of vesicles or RNA in vesicles, then you might avoid being fooled as you might be with TSG 101 or counting vesicles. And the other thing is it could make assessment or measurements more practical by allowing you to measure two RNA molecules with one platform instead of trying to measure protein in RNA or vesicles in some other way in RNA. In ongoing work in our lab, we are doing a prospective study of healthy volunteers, randomizing them to consume either a high salt diet first and then a low salt diet, or vice versa, to see if we can validate the findings that we have from the retrospective study that we did previously using banked samples. So this will be a new study. And we're also interested in sequencing the urinary extracellular vesicles using RNA-seq if that's possible. And we've seen some signals now from the group in Ghent and Munish Tiwari's group, a group at Rockefeller University. There's clues in those papers that have been published recently that make us think that there's probably some best practices. If you're interested in this problem, you want to talk about it with us, I'd love to discuss this topic with you of how to best sequence the long coding RNA in urinary EVs. And in other ongoing work, we are looking at patients who have diagnosed primary aldosteronism before and after they're treated with a mineralocorticoid receptor antagonist, or before surgical removal of the adrenal gland to remediate the problem, so that we can, because a biomarker of MR activation should change when you remediate the inappropriate MR activation. In addition, we are looking at the effects of a mineralocorticoid receptor antagonist on these biomarkers. The study design here is to take people with mild essential hypertension, wash out the blood pressure medication that they're taking, and then randomly assign them to take a mineralocorticoid receptor antagonist called eplerenone or placebo, then have a washout period, and then have them take the alternative treatment. And we'll collect samples and measure the biomarkers that we're interested in this setting as well. Of course, our work has been affected by COVID, as everything else has been, and we wrote about the considerations that prompted us to pause our work just before our institution asked us to. We're back to doing our work, and I'm hoping that we won't have to pause it again, although certainly the COVID pandemic is very active here. In terms of future directions, we've recently added Satish Rao, who's a PhD, to the lab, and he's added additional EV-related capabilities and experience. So we are really fired up about new clinical studies that we proposed recently for funding, and other ones that we'll be proposing in the near future. And just circle back for a second to COVID. We've recently done a blood pressure medication-related clinical trial in COVID, which we're getting published now. It's under review. And there are a number of other projects that I've worked on related to COVID. But the fact of the matter is, I can't wait to just get back to steady work on urinary extracellular vesicles. Some of the additional challenges I want to mention is we need to try to figure out how to make assays that can be used in practical ways. Some of the things we need to think about are pre-analytical variables that might be affecting our results. And thankfully, we have ISO working on roadmaps for the commonly assayed biofluids, including urine. ISO certification of methods would be good. I think I'm only aware of one ISO certified method within the world of EVs. It's a size exclusion chromatography assay, but I'm not aware of any other ISO certified methods. I think we need more of those. Automated methods, we need a lot of those, I think, because to the extent that they can be developed, certainly in clinical laboratories in the hospital, they don't want things that require a great deal of manual processing. They want things that are in cartridges that can be plugged into something and read and by a machine. Now, maybe the future of EV diagnostics will be send out laboratory work done in special facilities with high touch, very complicated processes like the exosome diagnostics model. I don't know the answer, but these are things we have to think about. Scalability is important. Can we incorporate robotics more into EV assays? Other challenges to be met. There's this constant tension between simplicity and purity. You know, when we're wanting to do biomarker work, how pure do our EVs need to be? And I guess it would probably depend on the exact constituents of those EVs and what non-vesicular impurities might be present in the preparations. And then there's this issue of openness versus commercialization. And I'm sure that we need both commercialization and the proprietary and closed concerns that come with that, and also more openness, more exchange of protocols, more exchange of pre-publication ideas, what we're working on, what we're thinking. I'm interested in trying to model that and also engage with others in that way. It's important, I think. So in terms of summary and key takeaways, interest in EVs as biomarkers in hypertension has been growing. To some extent, the methods have lagged behind the aspirations for now, and this has to do with funding and other things, but that's changing. Validation of currently proposed EV biomarkers in new studies is desperately needed, and challenges remain in establishing how we can perform these biomarkers in ways that are compatible with hospital laboratory workflows. I want to mention one more thing. In science, obviously, we build on the work of others, and as Newton said, if we have seen further, it's by standing upon the shoulders of giants. And if data underlying discoveries are not shared, it's more difficult to build on prior discoveries. And some people are doing a really wonderful job sharing research data. And we started an award called the Research Symbiont Awards to recognize those people and to encourage a cultural shift toward more data sharing. Actually, I'm giving a talk for the NIH tomorrow about this topic. If you or someone else that you know has shared research data in a way that is exemplary in your field, unusual in your field, I would ask that you please visit researchsymbionts.org to make a nomination. We would love to recognize and celebrate you or that person that has been a great data sharer. And because of sponsorship from the Wellcome Trust and a pediatric cancer research foundation called the Dragon Master Foundation, we're able to offer a prize that I think will mean something to the winners. So please check it out. Nominations are open until December 5th. And we will recognize the winners in a virtual awards ceremony in January that I think a lot of people will see. I want to thank the people who helped me do the work that I presented. Brian Bazell led a lot of the work that I showed you. Pradeep Gunasekaran played an important role in the lab, has recently gone on to do a PhD at Rutgers. Andrea Burrito is a very, very talented research coordinator who is running the clinical studies recruitment that I talked about earlier. And Dr. Satish Rao, we're very pleased to have him in the lab to help us develop additional assays and approaches for urinary EV work. And then we've got collaborators and mentors at the University of Michigan that have helped us get our work done. Dr. Scott Hummel, Bill Rainey, Rich Aukus, collaborators at Lanza, and Marco Bertini in Siena, Italy. Marco helped do the data analysis, the genomic analysis, and I want to thank our sponsors. And I want to thank you for your attention. And any questions you have, I'd love to take those now.
Okay, thank you so much,
My pleasure.
Brian. It's very nice presentations, and learned so much about the hypertension itself, the physiology of it, and then also how the EV can provide a biomarker for this disease. So I have a few questions, but I think I'll go with Phil first. Phil,
oh, okay.
please ask.
Thank you.
Hi, Jim. Phil Askin is at Yale.
Hi.
That's fantastic.
Thank
I
you.
wish you could infect the salt and water gang at Yale. I'll tell them about you. They don't know about you.
Thank you. I know one of the people there, and we're working on something. Well, we may work on something together. Yeah, yeah.
So I want to know things like, what's the donor cell of these EVs? What's the target cell? What percent of the urine EVs are the ones you're talking about? And there's a great assumption here that these mRNAs are working through DNA sequences. It's a huge assumption. You call them genes, and we don't quite know that. It's an assumption. And most molecules and old molecules have many actions.
Right.
So,
what a great series of points. So I think with respect to the cells of origin, that's a very good question. Well, I have found some interesting and surprising transcripts in the urinary EVs, and others have too. This is nothing special to me, actually. There are some things that one would think of as, let's say, preglomerular, probably, that seem to come from tissues or organs that you wouldn't necessarily expect to appear in the urine unless there was a mechanism for those RNA molecules to enter the urine from the blood. And I think how that could happen, well, it's anybody's guess. Some people have written very confidently that EVs are too big to cross the glomerulus. Other people have said, based upon the protein or RNA content of EVs in the urine, they clearly do cross the glomerulus. Until I see better evidence, I don't think either of those positions can be substantiated. Partly because there are many things that could happen. It might be possible that EVs are endocytosed in the proximal tubule, as many proteins are. So there could be an endocytosis mechanism, perhaps driven by megalin or cubalin. I mean, you can think of many different possible mechanisms whereby EVs somehow introduce their RNA content into tubular epithelial cells, and then somehow that is, transits those cells and ends up going from blood to urine, or either in a new vesicle, or somehow the vesicle perhaps could stay intact. Could there be some sort of paracellular passage of these molecules in between cells? I don't know. I think that we just don't know the answers to those kinds of questions yet. Go ahead. What do you think?
They're like, kids' balloons that can squeeze through and change so they can go through the glomerulus. I have no doubt about that.
Okay.
In your initial diagram, every one of those organs along the way is going to have an exchange of vesicles. And then we're finding that monocytes in tissue and fibroblasts in tissue are contributing important vesicles. There's a cloud of vesicles from all the different cell types.
That is very interesting. Very interesting.
I mean, that's many careers.
Many careers. I couldn't agree more. And some, when we did that comparison of the urinary EV transcriptome from the healthy volunteer with the different postmortem kidney cortex samples from GTEx, what motivated me to do this was I just wanted to know, as closely approximate what you were asking as possible. If you could understand what is transcribed at a given level in the kidney, is there any relationship whatsoever to what you find in the urinary EVs? Because that didn't have to be the case. It could have been, as you say, a cloud from everywhere. And it could have been that the whole signal falls apart and you can't infer anything about the state of the kidney. But that didn't turn out to be the case, I don't think. And you know, it's a simple analysis, but it suggests that there is a contribution of cells that are either kidney cortex or expressing pattern of genes that's similar to the kidney cortex to the mixture. But yes, it's got to be some kind of complex admixture. So then you could ask questions like, should you be trying to use pull down assays or something to enrich for the proper EVs that come from the tubular cells? Should we use?
We find affinity columns are a really good way to go.
Okay.
Even in affinity columns for the tetraspanins, you know, there's if a tetraspanin, it's a bell-shaped curve, and often the cells that are expressing the most maybe the subpopulations, and then there are other markers. But this is very exciting, and I love your passion.
Well,
you're going to find terrific stuff.
Well, thank you so much. I really appreciate that. I really appreciate that.
I'm a big fan of the tetraspanin itself, but I think something really a bit outside of it is that the Jenster group in the Netherlands, they're looking at urinary EV for prostate cancer biomarker. And what they've shown, interestingly, is if the patient had digital rectal examination, that increased the extracellular vesicles in the urine. So I think, I think there's some sort of like, you know, physical.
That's right. Yeah, others have published that too. And I've even seen a paper that sort of was touting the fact that they could see a signal that didn't involve DRE. I mean, people have been interested in that question for a while. That's a good point. Now, in the hypertension world, nobody wants to think about that. I'm sure. If the blood pressure is the issue, nobody wants to.
Yeah. Okay. Next question, Nikki.
But it's a very interesting point, and it does make you wonder if there are other physical maneuvers that could increase the excretion. I have actually heard that there are other things you could do that might jostle those vesicles free. I don't know if that, I haven't tried, but I have heard that maybe certain movements or something might make the vesicles appear more in the urine. I don't know. I'd be interested to know if anybody's published that, but that was sort of just things I was told. So.
Okay, Nikki, next question.
Hi, Brian. You did a great talk. I really enjoyed it. So question for you. So what starting volume did you use for your urine when you're doing your RNA assays?
4 to 5 ml.
Yeah, we had about 5 milliliters. So we had about 5 milliliter aliquot. And so I'm sure that we did subsequent work where we tried to quantify how many molecules are there. There's just not a lot of any given RNA molecule there. And so I think the preamplification insight, which was shared with me by Davida Zacco, I think that was extremely valuable and I would just say pivotal to being able to do this. I think later we understood through additional testing just how important that particular piece of advice was.
Do you see a difference between morning and fasting urine, Brian? No, not between, sorry, morning or fasting person, or is there a specific sort of criteria that you need to do to collect?
No, that's a good question. I don't think we know enough about urinary mRNA under different circumstances like that to answer those questions. We're doing dietary studies, but they tend to be focused on sodium, how much sodium the person is eating. But you can imagine there's a lot of studies that would need to be done to understand how homeostasis of these vesicles works. I mean, just a simple, there's some big picture questions that I have are, is the RNA that is leaving cells in the kidney into the urine, is it more or less a random sampling of the RNA that is inside that cell? Or in what ways is it biased? And we know with microRNAs, if you mess with YBOX1 protein, you mess with what microRNAs are packaged. And I know that there is a group at Harvard who said that there's a zip code to be found in mRNA that is packaged into EVs and that you can cause enrichment. That was one paper in, I think, one cell line. And I haven't seen anybody else say the same thing. So I'm not sure. I think that was Xander Breakfield's work. So I'm not sure, you know, the general impression I've gotten from watching a presentation that was given a number of years ago from George Church's lab at ICIV, and from the look at this question that we had, and maybe looking at the Ghent group's recent biofluid paper, I get the general impression that there's a pretty good survey of the RNA that's in the cell coming out in the vesicles. But I think more work needs to be done at the mRNA level. I think people have looked at it pretty extensively in other contexts like microRNA. But I don't think that people have done a lot of that with mRNA.
I wonder if there's a correlation between the protein that are found in the EVs and the mRNA, whether they are the same, you know, genes that are, and the same protein, not really genes, it's not the protein, but, you know, the same target.
Yeah, that's a good question. And because I think right now, correct me if I'm wrong, but we lack a mechanism of packaging. The thing that was interesting about that presentation from George Church's lab was that the person, Dima, who was doing that work, was trying to find a mechanism of packaging of mRNA. And it seemed that what he had actually demonstrated was that you just get a very nice representative sampling of what the parent cell looks like. If you look at, you know, in bulk, if you look at the bulk sequencing of the EVs, it looks like the parent cell. But he had done that in a limited number of cancer cell lines. He wasn't sure if maybe the packaging mechanism was broken. And I haven't seen it published. So these are just unpublished insights that I found interesting, let's say.
That's not true of microRNAs.
It's not true of microRNAs. Something different. The regulation seems different to me. As far as
the vesicles are very different than the donor cell for microRNA.
Yes. Yeah. But I'm surprised.
Vesicles that aren't even, they're hard to find in the donor cell.
Yes. That's what I have read as well. And I haven't worked on microRNAs, but I have seen the work presented that shows the ability to specifically manipulate what goes into a microRNA by messing with YBOX1 protein. I mean, it's clear that there is a mechanism of packaging. And
that there is at the target. We know nothing about what the vesicles do in the target cell and where and how they deliver what they deliver.
Well, one thing that bothers me is, let me ask you this. If you have some sort of epithelial cell that is secreting vesicles into urine, into, or, you know, just skin cells or something like that, does it not bother you that it seems like they're not going anywhere, they're just exiting the body? And maybe those don't have any, maybe some vesicles have a destination and some are just garbage cans or something. I don't know, but it's always bothered me that if you look at the EVs that are at the end of the urethra or whatever, they're coming out of cells at the end of the urethra, they don't have anywhere to go except out, right? I mean, as far as I could tell. Maybe I'm making a wrong assumption.
Well, there are epididymosomes, which was a field before any of us ever got going. And all along the pathway that sperm go, there are epithelial cells that make different vesicles to tune them up to make it through that tough oversight.
Very interesting.
Way ahead of us. Epididymosomes.
That is really cool. That is really cool.
All right.
This whole world is really cool.
Yeah, just a little comment that that's actually, I asked that questions to Harry Holthover, who's presenting at WebEVTalk, and he was mentioning there were some functions of urine EV actually, and one of them is related to the bacterial growth. I'm not really sure because it's not my expertise, so but it seems to be quite physiological relevance there.
Yeah, I know. I've talked about that with Harry, and I know that he has some interesting. He has an interest in microbiology, to some extent. And if you look at his papers about hydrostatic filtration dialysis, there is a theme in there about microbiology. It's quite interesting what he's found.
The bacteria have outer membrane vesicles.
Yeah, that's right.
Which is a lot of things are communicated between the bacteria and between the bacteria and the host via those vesicles.
Yeah, that's the cross kingdom stuff is just amazing.
Yes.
Mind blowing.
Yes, exactly.
Yeah, is there any other questions? I think I might close the sessions. It's a little bit over time, but we had such a nice discussion ongoing. So that was lovely. And thanks so much, Brian. And
thank you.
Yeah, for giving us a great presentation, so much details.